Okay. Well, thank you everyone for coming to our last but one seminar of the semester. It's a pleasure to have here Dr. Fitzpatrick from Duke University. He's in the Department of Neurobiology. Dr. Fitzpatrick is very renowned for his work in neural circuits as well as visual perception. His current work is on the state-of-the-art of optical imaging to probe the functional architecture of the brain. He's received numerous awards, among which the Alfred Sloan Research Award, the Cahill Club Cortical Discovery Award, and several others. He's affiliated with many prestigious centers, among which the Max Planck Institute for Neurobiology and the NIH. He's received numerous awards, among them teaching awards, and it's a pleasure to have him here. Thank you. The floor is yours.
Thank you very much, Mid. Am I allowed to move around? I don't have to be constrained to my chair there. Okay. I've had a great time this morning meeting some new people and talking about science. It's been a lot of fun. So my goal today is to give you some appreciation for the work that we've been doing that addresses how the cortex, as you'll see at the end, how the cortex wires itself. The processes that allow for neurons in visual cortex to develop very selective response properties that are absolutely critical for visual perception. And so I've divided the talk up into 3 parts. The first part of the talk is just a brief overview of the functional architecture of visual cortex and sort of the traditional view of how this cortical circuitry develops. And then I'll talk to you about work that we've published about a year ago, that talks about the fact that the selectivity for direction of motion, which is a property of neurons in visual cortex, is really unique in requiring visual experience for its expression. And the last thing I'll do is talk about some recent experiments that I'm very excited about, where we're actually able to visualize the emergence of this response property under the influence of visual experience. So let me start with just a brief overview. Are the lights okay for you? I mean, I guess we need the lights for, okay, fine. Yeah. Like a number of people that work on systems neurobiology, we're interested in the relationship between circuits. Would there be a pointer here by any chance?
Unfortunately.
Okay, that's all right. I'll wave my arms here. The relationship between neural circuits in cortex and perception. And as you'll notice, there's kind of a large gap in the middle there. And arguably, a major step in sort of filling this gap between circuits and perception was the discovery some years ago by Hubel and Wiesel that if you stuck electrodes into the cortex, you could identify neurons that would have very selective response properties. And one of the response properties they demonstrated was selectivity for the orientation of edges. So if you record from a single neuron, find its receptive field, and present gratings or line edges of different orientations, you'd find that a given neuron would have a certain orientation that it would respond maximally to. Now, what was also interesting about their work, and for the work I'm going to describe today, really critical, is the fact that there is an orderly columnar grouping of response properties in visual cortex. So that if you stick your electrode vertically into the cortex, this would be the surface of the cortex, this would be the white matter, almost all of the neurons that you would encounter along electrode penetration would have the same preferred orientation. And if you took the same electrode and arranged it tangentially, you would find in many cases, an orderly shift or progression in the distribution of preferred orientations. So, unfortunately, Hubel and Wiesel were confined to describing the mapping of orientation with an electrode, which is a very difficult way to build up a 2D representation. This awaited techniques that would allow you to visualize the distribution of activity in the brain when you're presenting a given stimulus. And one technique that we've exploited that was actually generated by two people, Miriam Grinwald and Gary Blaisdell, is what's called intrinsic signal optical imaging. And intrinsic signal imaging takes advantage of the fact that there is a change in the hemodynamics that are associated with neuronal activity. Those of you who have worked with fMRI images, functional fMRI images, the signal that we're looking at here is very similar to the signal that would be found with functional MRI. But here we're using light to visualize it. So we're illuminating the cortical surface with red light. We're looking at changes in the reflectance of light from the surface. The changes in reflectance correlate with activity. So when you look at these images, here you're looking down on the surface of the cortex. This is the visual cortex in an animal. This is the ferret, and I'll tell you more about the ferret in a second. You see these dark patches. These patches, if you were to stick an electrode in those patches, you would find that those patches contain neurons that respond selectively to a horizontal edge. Now, the beauty of this technique is that I can present many different stimuli, look at the same bit of cortex, and I can really understand how this property of orientation is mapped out over the cortical surface. And that's illustrated in the next slide. So what you see here are individual images, and each of these images was collected when the animal was viewing a grading of a different orientation, horizontal, obliques, and vertical. And then what we can do is quite literally build up a pixel by pixel map of this property of orientation selectivity. And you can do this in a variety of different ways. You can literally create tuning curves for individual pixels. You can do a vector summation. You can do a polar analysis, etc. But effectively, what you can do is find out which of those orientations was most effective in modulating the reflectance from this particular bit of brain. And what you end up seeing is quite literally a beautiful map. It's an iterated map because the same orientation will appear multiple times across the surface, but nevertheless an iterated map, a systematic map of this response property. Now,
this is just purely at the surface.
Yes, so you're imaging at the surface, but you may remember the electropenetration that I showed you. So the beauty of this all rests on the fact that you do have this columnar arrangement. So what you see at the surface also characterizes what you see deeper at that point.
Yeah, but your measurement equipment only captures.
We're capturing what's happening at the surface,
okay.
But that's again reflecting what's going deeper.
Okay.
So now, orientation isn't the only property that's mapped in the cortex. Our vision would be pretty limited if that were the case. Another property is direction of motion. And so you can use the same strategy. So you can present, for example, a horizontal grading. You can move it up. You take the same grading, move it down, and then subtract the images of the cortical surface for those two different stimuli. And you can identify hotspots, these black regions here, which are the regions that contain neurons, in this case, that prefer motion upwards. And the black regions here would prefer motion downwards. And you can use the same strategy, again, either creating tuning functions for pixels or doing a vector average. You can come up with a map. In this case, this is a map of direction preference. Now, the obvious question that you would one of the obvious questions that you would raise is, how are they related to each other? And we have probed this, others have probed this as well, and there's a really nice systematic relationship between the maps of direction and the maps of orientation. So if you find regions of the map of orientation that are changing relatively slowly, so here's a region that prefers near vertical, you'll see that it can be divided up into two sectors that prefer opposite directions of motion. So quite literally, there's kind of a place map, if you will, for these properties. And if you knew, for example, that there was activity at this location, you could do a pretty good job of predicting what the orientation and the direction of motion was, provided you have a limited range of stimuli. Okay? Now, this extends further. There's orientation, there's direction, there's what we call ocular dominance. So the influence of the two eyes can vary across the cortical surface. So in some regions of the cortex, one eye will dominate. Other regions, the other eye will dominate it. So it's a sort of like a tessellated pattern of dominance by one eye or the other. And there's also a map of visual space, which I'm sure you're all familiar with. So you can you get the sense that there are indeed multiple maps that are superimposed within the cortex, and recognize that each individual cell can possess all of these properties. So a single cell will have a preferred ocular dominance, it will have a preferred receptive field location in space, it'll have a preferred orientation and a preferred direction. Yes, sir?
You just answered my question.
Okay, great.
But the ocular dominance is valid for all instances. In other words, it's not scene specific. It's going to be, for instance, if it's the right eye, it's going to be that region for the right eye all the time?
Yes.
Okay.
Yeah. These are, I wasn't going to go here, but I will tell you that this is the classic picture of maps in cortex. We published a paper in 2003, it's Bezzoli et al., where we really challenge this idea. And I'm not going to go there today, but if you're interested in it, what we end up saying is this notion that this bit of cortex represents the intersection of feature maps is way too simplistic. And there's a much more interesting way of thinking about it in terms of spatiotemporal energy filters. But I'm not going to go there today. But I actually get very excited talking about that because it's a very different way of looking at the same bit of brain. So, having said this, and I've emphasized some aspects of the systematic relationship between these maps, and if I went further with that, the ocular dominance patterns are also systematically related to these other mapping properties, and you get the sense that considerable amount of energy has been invested in making this system very precisely wired. And given that, the title of this paper, you can imagine this was not something that made my day when I saw this published, a cortical column, a structure without a function. And what this paper says, and I have to admit that if you push on me, I can't rule out the possibility that columns are epiphenomena. But what this paper basically says is it reviews the literature, and it takes advantage of the fact that there can be differences between species in the kinds of columnar structures that are present, or whether these columnar structures are actually present. And in some cases, even within a species, there can be animal-to-animal variability in the columnar systems. Now, the problem is no one has been able to find a difference in perceptual capabilities that would accord with these differences in columnar structure. So, for example, rodents do not have orientation columns. And you'll say, well, of course, a mouse, a rat, what do they see, right? I mean, they can't do very well. But if you take an animal like a squirrel, like a gray squirrel, its visual system is really very, very complicated. It's invested a lot of energy in that. Its perceptual capabilities are very high, and yet it doesn't have an orientation column system. So when I saw this, I must say that this was my take on this. A cortical column, a structure without which I would have no funding. And I quite literally mean this, because if you look at all the stuff that's come out of my lab, in the last 10 years or so, it's all based one way or another on using these functional maps. And my counter to that title is cortical column structure critical for probing cortical function and development. So even though I can't articulate exactly what the function of a cortical column is, what we've done, and others as well, is use this columnar system as a functional referent. You know, as I said, we know that when we stick an electrode down into this location, what the properties are that we will encounter at that location. So now, if you're interested in what are the rules of connectivity in this network, well, you can do beautiful experiments where you make an injection of a tracer substance and look to see how the axons that are associated with those neurons are labeled and how they distribute relative to this pattern. So you can know, for example, do they contact cells that have the same orientation preference or a different orientation preference? Where are their receptive fields located in visual space, et cetera? And a lot of the stuff that we've done has taken advantage of maps to do that. We've also looked at population code for stimulus features. That's the Bizzole et al. Paper that I talked about, where once you know what the mapping structure is, you're free to present a variety of different stimuli and ask, how is that encoded by this network? And if you didn't have the functional referent of knowing what the cells were at that location, you would really be stuck. And finally, it's been actually, invaluable in terms of looking at how the circuits develop. So even if I can't tell you why you need columns in terms of how the visual system functions, I can tell you that without them, I wouldn't have much to talk about. It's basically made it possible to dive into different levels of function that otherwise we wouldn't be able to look at.
Another question.
Yeah.
So when you show this, though, for instance, for vertical or whatever direction you want, these are the responses that you have are sort of varying degrees, though, right? It's not an absolute. It's not
sure,
an on and off thing. Right?
There's a tuning function. So if I look at the neurons that are sitting here, their peak response will be vertical.
Right.
But they'll respond, say, plus and minus 20 degrees of vertical, and nice sort of Gaussian function.
Some response from the others to a certain extent. No.
No, the tuning it's tuning, but it's not so broad that you would get a response to the orthogonal orientation.
Okay.
You don't see that. It really, the tuning is, as I said, plus or minus 20 degrees, you would get significant response, and then there would be, but orthogonal, no.
Okay.
Yeah. Okay. So what I want to do is to focus on this part of the talk, and in retrospect, I admit it might have been more interesting for you guys if I had talked about the perceptual side of this, but the stuff that we're doing with two-photon imaging is so cool that I thought you'd get a kick out of that. And so the starting point for this is really this question that it's one of those things that magazine was sitting on the coffee table and I couldn't help myself. Here's the question: how much does experience matter? And as I look closely, it even says why science says it may be overrated. And this actually rings true, not with. Okay, I'm not making any statement about politics here. I really am not. But this is actually an important issue in terms of the development of cortical circuitry. There's how science kind of swings, right? And there was a period of time when people thought that really the wiring of cortical circuits was largely dependent on experience. And the pendulum has swung to the point where I think if you asked most card-carrying developmental neurobiologists, they would say it's molecular cues that are guiding the construction of cortical circuits. And, experience plays a role later on, but in actually building the network, it's a molecular guidance problem. What I'm going to argue today is that that's true for some features, but not others, and that direction selectivity is really unique in this regard. Now, here's the sort of typical storyline, and I'm showing it here in the context of the animal that we use, which is the ferret. And the ferret is it's a carnivore, and in terms of its brain structure and whatnot, it's kind of like a small cat. And one of the advantages of using the ferret is that it's born in a relatively immature state. So here's date of birth. And you can see that there's almost a month that transpires before the eyes actually open. And you can see when different components of the visual system become active and when they arrive. These are sort of the milestones in development here. And what I'd like you to do is to think about those columnar systems that I mentioned to you. And what you're seeing is the traditional way of subdividing this into experience-independent emergence and then experience-dependent refinement. And I've plotted out what people think about ocular dominance columns, and the idea is that they actually form very early on in development before the eyes open. Now, those of you who know ocular dominance plasticity and critical period and whatnot, these are things that occur long after the eyes open. And you may know the famous Hubel and Wiesel experiments of suturing one eye and seeing the cortex basically become dominated by the open eye. These are events that occur way out here long after the circuitry is actually set up. This is true for orientation as well. So if you do experiments where you literally go in and you open eyes prematurely, you can find that there is orientation selectivity present, so it doesn't require experience. Nevertheless, if you were to rear an animal in an artificial environment where all it could see was a certain orientation, you can change the structure of the cortex. And finally, here's the question. What about direction? Is direction the same as the... I will say that it wasn't as if we just went into this blindly. We realized in the literature that no one had actually addressed this question. And selectivity for direction of motion is actually non-trivial. Because what you're doing is you're building a receptive field that's sensitive to both space and time. So there are a number we could talk about mechanisms that are used to set up direction selectivity, but that's not something. Orientation is totally spatial. Ocular dominance columns, this is really axon guidance from the lateral geniculate nucleus to cortex. It's not a big problem or a hard problem to solve. Direction selectivity, where you have to make a neuron selective to the correlation in space-time that represents a direction of motion in space. That's a little more difficult. In fact, the modeling community, to do this, they've always used a training stimulus. Now, that training stimulus could be the environment, or it could be waves of activity in the brain. But it was something that couldn't be done without having some sort of a training stimulus. So the starting point for this was simply to ask, what do we see at the time of eye opening? And that was our clue that there was something interesting going on here. So let me tell you what these are. These are maps of the cortex, and this is orientation being shown here and direction being shown here. Okay? This is the same cortex being imaged for different properties. And this is postnatal day 30. First of all, you see orientation is present at all times. Direction is not. Direction is not present early on, and then begins to emerge here and then becomes stronger with time. And this is illustrating, this is our metric for the magnitude of these properties, and the red line here shows you that indeed orientation is there and grows, where direction, you don't start seeing it until after eye open, and then it rapidly grows and becomes mature. So that suggested that there was indeed a difference between these two properties. Didn't say that experience was required. It could simply be that direction emergence is delayed relative to orientation. So the experiment to do to test that is to ask what happens with dark rearing. So you can rear an animal in the dark. I mean, we're not rearing them for very long in the dark, actually, but bringing them out at these different time points. And you can ask, is direction there? And what you see was very clear. Now, for some reason, I didn't realize that it got in reverse, but direction is being illustrated here, and orientation is being illustrated here. And what you see is zip on direction, but very clear orientation selectivity. So again, consistent with the idea that not only is direction selectivity delayed in its emergence, but it requires experience for its expression. This may be obvious to you, but if not, I'll just emphasize it. The point is that it's not that the cortex is unresponsive in these animals. The cortex is responsive, otherwise we wouldn't see orientation. The point is that it's responsive and it can respond to orientation of edges, but there's no selective response for direction of motion. So there aren't neurons that are selectively responding to one direction versus another. Okay, trust me, if you stick electrodes in the brain, you collaborate what you see with intrinsic signal imaging. Now, the final point that I'll make with this is that you might imagine that it doesn't matter when the experience occurs. You know, maybe if you delay experience out here to in the 60s and give a couple of days of experience, that would be sufficient to recover this response property. And what you see, so if you dark rear beyond the time period when this property normally emerges, and then give experience for 3 weeks, and you compare that with what happens if you dark rear until the time where it just emerges and then give experience, is very different. So again, here's what the normal direction map looks like. Nice modulation. Here's what a dark reared animal looks like. I've already shown you this. This is what happens if you dark rear until postnatal day 45 and then give 3 weeks of experience. No recovery. And then finally, this is dark rearing until just before the time that this property normally emerges, and then 3 weeks of experience, and then you get the pattern. And this is just showing you this result for multiple animals. So the first part of this talk, what I hope... Yes?
So in the previous slide, when you say that there is no response if we dark rear until 45 and then still do it, does that mean those neurons are not used for direction, but then they might be used for something else?
Okay, great question. So, all of the neurons, I can't say all of the neurons, but the 2 photon data suggests that this is true. They remain responsive, and they're selective for the orientation of edges, but they just don't show a differential response to direction. Now, you could ask, what are the other differences in the properties of those cells? And we've explored it, that's a large parameter space. We've explored it in terms of looking at spatial frequency, temporal frequency, contrast sensitivity, etc. And in animals that have, basically gone through this, so they've been dark reared during the period where direction emerges and then given experience, all of those other properties normalize. So you wouldn't be able to, based on those properties, you wouldn't know that those cells were any different than what you would see in a normal animal. And the only thing that seems to be missing is this property of direction selectivity, which was actually reassuring to us because you can look at the paper. This is a Nature Neuroscience paper from a year or 2 ago. What's nice about it is you can say it really is selective. And you could imagine changing the spatial temporal properties of the neuron, and for that reason, not see direction selectivity. But the fact that all of those properties normalize and direction isn't there says that somehow you've managed to alter the network in a way that prevents its expression without altering these other sort of fundamental properties of the cell's receptive field. Okay. So let me just sort of conclude with this first part. They emerge later. The direction columns come later. It looks like you need early experience to get them to emerge. And that requirement is unique to direction selectivity. And thanks to your question, I've actually covered the data to support the last statement there. Now, I would be the first one to admit that dark rearing is a pretty blunt tool to probe the role of experience. And what you would like to know is experience with motion in the visual scene important for generating cells that are tuned to that response property? And obviously the dark rearing doesn't get at that question. And we wanted to find a way to do this, to ask, does experience with moving stimulus induce the emergence of direction columns? And we came up with what turned out to be a really simple paradigm to explore this question. So what we did was we took a visually naive animal, which had no direction columns, set it up for imaging, looking at a screen, and at the start of the experiment, we took recordings of the cortical surface, and so we knew we had orientation, but we had no direction. And then we continued to train, and we trained by just taking the stimulus and moving it back and forth. And at different time points, we would look at the cortical surface to see whether there was any sign of direction selectivity. And when we started out, there was basically nothing. And then slowly but surely, we started to see the pattern, the selective pattern, emerge. And we could start to see a hint of it maybe around 8 hours or 10 hours. But then it get progressively stronger and stronger up to 18 hours. Now, I would acknowledge that the strength of this pattern is not, it's nowhere near what you would see in an animal that was mature. But nevertheless, the differential signal here was robust enough for us to quantify. And now, the graduate student who was working on this project, his name was Ye Li, had to face significant skepticism here, right? So this makes it look like the animal has acquired direction selectivity as a result of the training. But when you come down to it, the way this experiment is designed, we don't really know that the pattern that emerges here is really a function of the stimulus that's being presented. You could imagine that there are general changes in cortical physiology or something over this time course that would lead to the emergence of direction domains. So luckily, students smart. So, well, I've controlled for that. Oh, what's that? Well, at the end of the experiment, I can test multiple directions. And I can ask whether I see direction emerging just for the training stimulus or whether I see it for all of the directions. And this is what you see. So in this case, the stimulus that was used was a horizontal grading. That was the training stimulus. And you see direction emerging for those directions, but not for the other directions. And here are 3 additional cases, all of which show the same phenomena. So no matter what the direction of the training stimulus was, that's what gives rise to the direction domains, and you don't see it for others. Now, here's just the quantification of this for a number of animals, showing that you get significant signal for the trained directions of motion, comparing the start and the end of the experiment. But you don't get it if you look for directions that are orthogonal to the trained direction. And actually, I'm not going to, there are other controls here to rule out an increase in the activity associated with orientation and whatnot, but I won't go into that detail. So let me summarize this part, which is to say that what we've demonstrated is that exposure to a motion training stimulus produces this rapid emergence of columns. And I will say that this is surprising. If you were to look at the literature, you won't find examples of papers showing this kind of rapid change in cortical response property. The effects are limited to the columns that are activated by the training stimulus. This is key for telling us that it really is, in a sense, learning from the presentation of the stimulus. It's also key because it tells us that this is happening, at least in part, at the level of the cortex. Cortex is the first point along the visual pathway where you have cells that are tuned to the orientation of edges. Okay? So it can't be something I mean, it could be that there are changes in the retina, could be there are changes in the geniculate, but that can't be the explanation, or at least the complete explanation for the phenomena, because we see this orientation specificity. And it occurs without increasing the magnitude of response to the trained orientation. It's not just some sort of a thresholding phenomenon that's associated with the orientation of the training stimulus. I'm sorry, you had a question.
Yeah. Excuse me. I assume you locked the ferret's head so he couldn't turn. Now, if you let him free, I just wonder if he would educate himself for both motions. You know what I mean?
Yeah, we have not done that. So the animal is for imaging purposes and others. And also, these animals are, they're paralyzed, so they can't move their eyes, which can be another problem in terms of torsional movements and whatnot that could create issues. But I should emphasize something here, and I'll come back to it when I start talking about the 2-photon data. These guys are not getting just one direction of motion. They're getting two. They're getting opposite directions of motion. And somehow the cortex is sorting this out and producing a differential pattern. So one question that you might be asking yourself is, what's the information that tells this neuron to prefer that direction of motion, and the neurons in a cluster next to it to prefer the opposite direction of motion? And keep that thought. It'll come back in the 2-photon data. So I'm sorry, did I answer your question? Okay. So this is intrinsic signal imaging is very useful. It gives you this kind of a columnar view of cortical activity, but it's activity through the filter of hemodynamics. So we really don't know what single cells in these regions of the brain are actually doing and how they're affected by this training. Now, it turns out that there is a relatively new technique that allows us to get at that. So we can visualize the activity of single neurons. This is called in vivo 2-photon imaging. And this is a technique that's only been available for a couple of years now. And quite literally, you put the animal under a 2-photon microscope. And so this is, you're not seeing the microscope here, you're just seeing the microscope objective. Light is coming in, and it comes into the brain. It takes advantage of what's called the 2-photon effect in physics, where basically if you have pulses of light with a really high density of photons, at twice the wavelength that's necessary to activate a fluorescent molecule here. Because of their density, occasionally two of these photons will collide, and it creates a single photon of half the wavelength. Now, the beauty of this technique is that it tremendously reduces light scatter. So you only get back those regions of the image that have had this 2-photon event occur. And what it allows you to do is to literally take very thin sections through the cortex and look at the distribution of fluorescent tags. Now, in this case, what we're doing is we're looking at a marker for calcium. It's called Oregon green bapta. And so we're looking for changes in fluorescence that, that signal changes in calcium levels. And it turns out that calcium levels are tightly correlated with spike rate. So neurons that are spiking have increased calcium levels. Now, I'll show you what it looks like to image with this technique. This is just the anatomical images here. And what we're doing is coming from the surface down into the brain. So that's the surface image. And you'll see as we go deeper and deeper that the density of cells actually becomes greater. And that's not because we're superimposing cells. That's just the way the cortex is structured. Layer 1 has very few cells, And then layer 2, 3, the density of cells becomes greater and greater. Now this is just a stack of images taking you deep into the cortex. We can go about 300 microns into the cortex. But you see the beauty of this technique is that you're literally visualizing individual neurons as opposed to the intrinsic signal imaging technique where you're looking at changes in hemodynamics. And here's what it looks like. When you're actually activating the cortex. So what you're seeing here is the presentation of two different visual stimuli. I said activating the cortex. You realize that we're presenting gratings to the animal, which is stimulating the retina, which is ultimately activating the cortex. And what you're seeing is cells light up. And you'll notice here are the two different angles that are being presented. And you'll notice that sometimes you get cells that are lighting up down here, and sometimes you get cells that are lighting up here. And you're literally visualizing the columnar structure of the cortex at the level of single neurons. Okay? Now, what can we do with this? Well, on a good day, when things are working, you can go in and assess what the orientation and direction selectivity is of individual neurons in the image. And this happens to be from a visually naive animal. And the lines here represent the preferred orientation of the cell at that location. And the arrows here represent cells that had a direction selectivity greater than 0.5. And 0.5 for us means they respond twice as well to one direction as to the opposite direction. And that is usually the threshold that's used to call a cell direction selective. So what you see in a visually naive animal is what you might predict from the intrinsic signal imaging. So cells that are responsive to the orientation of edges, but very few of these cells give rise to a direction selective response. Now, this is just to show you our metrics. I don't know how well this shows up, but basically this is our direction selective index for cells. And this is showing you the tuning curves. You'll notice that all of these have two peaks because we're dealing with direction in 360. So the two peaks then represent opposite directions of motion. And you know, they can be basically equivalent peaks where the direction selective index will be close to 0, or you can have a single peak, in which case the direction selectivity is close to 1. And this is what you see when you compare a visually naive animal with an animal that's had experience for 7 days. And so what we're seeing is that there's a transformation here. You have cells that basically aren't very responsive to orientation becoming, aren't responsive to direction of motion becoming much more responsive to direction of motion. And you can see the distributions down here, and they're for sure significantly different. Now, what we wanted to do, and this is where it got, it helps to have a graduate student who's got, I say he's got golden hands. He's really, really terrific. What you'd want to do is to be able to follow individual cells during this training procedure. And so this turns out to be non-trivial. And I won't go into the details of why it's difficult, but one of the reasons is that the dye itself is fading over time. And so you're limited in how much information you can get out. But the experiment I'm showing you here, we have cells before training, and here's 2 cells in this field that we've identified. And just because you can look before and after training, and you can use the position of the cells to make sure that you know which cell you're looking at, you can follow these cells over time. This is what their tuning curves looked like before the training, and this is what their tuning curves look like after the training. And of course, I'm pulling out 2 robust, very prominent examples, but we see exactly the same thing in multiple animals, although the effects are not necessarily as great as the ones that I'm showing here. But clearly, these cells have changed from not responding in a selective way to direction of motion to illustrating direction selectivity. Just to show you some other examples, here are here's the before and after. This animal was trained for 10 hours. Again, showing you a number of these cells go from not showing a selective response to direction of motion to exhibiting one. Oops. Here's another one, before and after. And what you can do, of course, is for each cell, you can plot its direction selectivity before training and its direction selectivity after training. And you end up seeing that there are indeed a number of cells that show this increase. Now, somebody should be saying, how do you know it's related to the motion of the stimulus? What if it's just activity? You give the animals activity and this occurs. So the experiment that we did to control for this is what we call flash training. So instead of drifting the stimulus, we flash the stimulus at different phases. Okay? So even with a flash, those of you who know spatial-temporal energy know there is some motion energy there, but it's nowhere near what you have with the drifting grading. And what we saw was no significant change as a result of the flash training. And this kind of summarizes all the experiments that I've shown you. So cumulative percentage of cells in experienced animals, this is showing the DSI, and in the visually naive animals. So, I mean, it basically shows you that the median value here in the naive animals is about a 0.1, whereas in the mature animals it's up close to 0.5. And if you look at what's happening with training, training is somewhat intermediate between those two. And this is what the, I'm sorry, that's motion training is the red line. And flash training basically is no different from the visually naive animal. So everything that I've talked to you about up to this point with respect to the two photon work has been magnitude of direction selectivity. So it's asking, do the neurons become more selective? What I haven't addressed is whether these neurons change their preference as a result of the training. And so what you'd like to know is, is the preference predetermined, and all the training does is increase the magnitude of that initial preference? Or does the training lead to changes in the cell's preference? Now, what I've done here is to show you this map. And instead of thresholding the map, I'm showing you the direction preference associated with each neuron in the naive animal. And the direction selective index here is being shown in a color-coded fashion. So this is the visually naive animal. Not surprisingly, they're all down here on the blue end of the spectrum. All right, magnitude is low. And when you look at the same neurons after training, the magnitude goes up. Again, that's totally consistent with what I've shown you. But if you notice, there's another thing that happens here, and it's hard to see given the lighting and whatnot. But some of these neurons actually flip their direction preference. And I've circled them here so you can see it. So all the ones that are circled here, if you watch, you'll notice that not only do they change in their color, which means they're getting stronger in terms of their direction selectivity, but they're also changing in what direction they prefer. Okay? Now, if you look closely, there are some, the ones that are circled, that change to prefer motion to the right. And in fact, most of them do that. And there are a few that actually change to go to the left. Now, when we saw this, we got very excited because this suggests that not only is training increasing the magnitude of direction selectivity, but it's actually impacting the preference that the neurons are exhibiting. This gets tricky, however, to define, and you would probably understand the reason. Because initially, the selectivity is weak. And so you want to know, is that really a change in direction preference, or is that just because that initial direction preference was so weak and our ability to measure it is poor? So I'm blessed with a postdoc, a former Caltech undergraduate engineering student who says, we'll do this with Bootstrap and we'll pull this out. And what we've done is come up with a way of quantifying the degree of organization that exists in this pattern, and then to ask whether these neurons, we can say with any certainty that these neurons are flipping their direction preference as a result of the training. And the first step in this is basically to define a coherency index. So what we do is take for each neuron, we look at the neighbors to that neuron, and we talk about its coherency. If all of the neighbors are pointing in the same direction, it would be 100. If all of the neighbors are pointing in the opposite direction, it would be minus 100. And of course, if there's no particular relationship, then it would be 0. And when we look in the visually naive animals, and we look at that local coherence index versus distance, we do see that there's a little bit of coherence. So on average, even in the naive animals, a neuron is a little bit more likely than not to have a neuron that has a similar direction preference. This falls off with distance, as you would expect for something that's mapped. If you look in mature animals, there's a whopping amount of coherence. Okay? So what you see in the immature animals is nowhere near the coherence that you see in mature. And what you see with training is in between. So the training leads to an increase in the degree to which neurons are like their neighbors. So now let's get back to the switch. Yes?
Why does the green line turn back up at the end?
Great question, and I don't know if it's included on here. Yeah. So these points happen to be significant. That one is not. So what you'd like to know is what's accounting for this increase in coherence. And one possibility is that it's the neurons that are actually flipping in their preference. Now, if that's true, then there should be a relationship between the initial weak map and the probability that a neuron would flip. So if you're a neuron and you're sitting in a part of the map where you're like your neighbors, then you're expecting not to flip. If you're in a part of the map where you're not like your neighbors, you'd expect to see a higher probability of flipping. And that's essentially what we see. So if you look at the probability of the preference reversing versus the local coherence before the experiment, if you're a neuron that's in a region that has positive coherence, you're less likely to flip. If you're in a region of the map that has negative coherence, you're more likely to flip your direction preference. Now, I submit that this is actually really interesting and very different from the way experiments are generally done with experience. Most of the time, these experiments are done by taking an animal, presenting one direction of motion, one orientation, and you see that the cells mimic the stimulus conditions. That's not what's happening here, because we're giving these cells the opportunity to respond to two opposite directions of motion. And they're sorting this out to produce this orderly map. And we'd like to suggest that we're looking at the product of the local network with Hebbian mechanisms building its own structure. So there may be an initial seed that's planted prior to experience, But that initial seed is elaborated on by experience to build this nearest neighbor relationship. So let me just, yes, okay, so obviously you want to know whether this happens without the motion training stimulus, and it doesn't. So if you just flash the stimulus, you don't see this sort of self-organization property. So what have we learned from the ferret? Well, just to summarize the last part of the talk, the single neurons in visually naive animals have weak directional biases, and they don't have the strong local coherence that's characteristic of a map. The training with a motion stimulus strengthens the responses of neurons, and it can maintain or reverse the direction bias in a fashion that builds local coherence. And finally, the big take-home is that experience with moving stimuli plays a critical role in the early construction of the circuits that represent direction of motion. And I've mentioned, I think I've mentioned Ye Li a couple of times, maybe not by name, but this is the graduate student with golden hands who actually defended just last week and did very, very well. Another collaborator on the project is Steve Van Hooser, who's really responsible for all the two-photon imaging that I've presented. Mark Mazurek has helped us out. He's also another postdoc tremendously with the quantification of the two-photon data, which turns out to be quite a bear, actually. And Len White is a colleague in the physical therapy department that I have been collaborating with for years on all our stuff developmental. And the rest of the people in the lab have contributed to discussions of the work that I just presented. So on that note, I'll stop.
Any questions? Yep.
So for all the experiments, does the result change over the age of an animal?
So unpack that a little bit so I know exactly what you mean.
So for example, if you found that neurons in this area are responsive to this kind of direction or orientation, but then over a period of time as the animal ages, does that pattern change?
No. So once, well, let me see. So if you were to image the same animal at multiple time points, and we're talking about a mature animal now,
right,
you would see the same pattern. It would not be altered. Now, if you deprived the animal in some way of normal visual experience, you could change the representation in the cortex, but only during the first month or so after visual experience begins. But you could, for example, bias the cortex to represent some directions more than others. Another question that I'm frequently asked when I present this is whether it's the case that all orientations and all directions are equally represented in cortex, and they're not. So it turns out that cardinal orientations occupy a larger proportion of the cortical map than oblique orientations. And this corresponds, some of you may know the perceptual stuff behind this, but you're much better at making discriminations of small angular offsets for cardinal angles, particularly, actually I think it's horizontal that you're better than vertical, but both of them are better than obliques. And presumably it's related to how much of the cortical surface area is occupied by neurons that have a given orientation. In fact, there's a correlation with natural scene statistics as well. How much of these various orientations are present in natural scenes.
And again, do these neuron maps change between different animals of the same species, or do you don't see any variations? Say regular, normal growing animals.
Yeah. So I think what you're asking me is how stereotyped the maps are in different individuals.
Right.
Yeah. They share certain features in common. So the average repeat distance in the map, for example, will be similar between animals of the same species. In terms of the biases that I just mentioned, that kind of thing is consistent. But and there may be some features. So, for example, if you get near the boundary of visual cortex, many times the what they're called iso-orientation contours tend to run perpendicular to that boundary. And so if you looked across individuals, you would see those same features. But if you actually looked at the structure of the map, so for example, if you looked at where every pinwheel center was, or you looked at the progressions in a particular region of the map of visual space, those would be different. So it's as if the basic outline of the map is preserved, but the details are not.
Other questions? I have a question. I guess what this makes me think of is perhaps when you were talking about the ocular dominance and things like that. Has it been established perhaps, or has anybody looked at that? Does that differentiate perhaps then the development of such areas among perhaps more or less artistically oriented people? And
so as you're getting to the question of can you predict visual capabilities based on underlying structure in visual cortex and stuff like that. There are, I'm going to leave vision for a moment just to make the point, and the reason I'm doing that is that I would say that correlations between the structure of visual cortex and performance they haven't been well worked out, at least as far as I know.
Mm-hmm.
But if you look at the auditory system and you take people, for example, who are musicians or people who have perfect pitch and so on and so forth, you can find correlations in the amount of cortical surface area that's devoted to representation of frequencies that are relevant to what the artist does and so on and so forth. So there's no question that in some cases you can find those correlations, but with respect to visual cortex, I'm unaware of a direct linkage like that.
I was a question along those lines as well. I'm not familiar with the area, so I was curious about this. When you talked about the experience of suturing one eye, you mentioned that the ocular dominance changes.
Yes.
And earlier than that, you mentioned that the cortex is a tessellation of right and left dominance. Does that make the cortex like a Swiss cheese if one eye is shut off?
No, and just the opposite. So, and this only happens if you do this in young animals. There's actually a critical period of time where you have to do it. Those neurons then become responsive to the open eye. So it's not like I assume by your question you meant that maybe there are holes where it's not active. No, in fact, the beauty of this is that you get to use that circuitry and keep it active, but now it's devoted to processing information from the open eye, not from the closed eye. Now, I think it's an interesting question. What would happen if you did that in a mature animal, where you don't have the plasticity that's present in young animals? If you looked in layer 4, which is where the initial inputs terminate, there you would basically see regions that were not active and then regions that were active. So without plasticity, you wouldn't get this filling in.
Nice.
But early on, you would fill in.
Okay.
Okay, thanks very much.
Thank you.